Good morning, children. Today we will be discussing about the regulation and expression of gene in prokaryotes. Now, see children, before we begin with the prokaryotes, let us summarize the gene regulation in case of eukaryotes. Now, as I told you that in case of eukaryotes, there is DNA, then mRNA, and then protein, and this is called as transcription and this is called as translation. Now, everything I have already taught you. Now see, in case of eukaryotes, where all these processes can occur, the genes can be regulated and expressed at the different levels. For example, when this mRNA is being get prepared or informed by the DNA, that is, if the DNA helps in the formation of mRNA, at that time the genes can be regulated and it can be expressed easily. Clear? So the meaning of regulation and expression is the gene has to check everything, that whatever process is going on, whether it is conducting in the proper way or not. This is the job of the gene. Okay, whether the information that has to be transformed to tRNA and rRNA for the protein synthesis, whether that information is correct or not. So this is called as the expression of the gene and the regulation of the gene. Okay, so in case of eukaryote, this is the first step where the gene can regulate all the expressions of the mRNA. After that, when this mRNA comes out of the nucleoplasm, when it goes to the cytoplasm, where it can combine with the tRNA, okay, where tRNA can encode all the information, or when tRNA transfer all these amino acids to the rRNA for the synthesis of the protein. So these are some specific points where the expression and regulation of gene can be checked. Clear? But in case of prokaryotes, now our topic is gene expression in case of prokaryote. In prokaryote, the important factor is environmental factor. Okay? This environmental factor affects the regulation and expression of the gene. After that, one more point, and it is availability of resources. Okay, availability of resources. So these two are the important factors in case of prokaryote which can regulate the expression of the gene, that whether the gene is expressing itself properly or not. Along with this, one more important point is there, children, and that is accessory protein. Okay, this accessory protein plays an important role in case of prokaryote to regulate the expression of the gene, whether the gene is working, expressing itself properly or not. So it is controlled by accessory protein. Okay, now children, this we have to see in detail. In case of prokaryotes, multiple genes are formed on one segment. Okay, what I am saying, in case of prokaryote, multiple genes are found on one segment of DNA or RNA. Okay, so on one segment of RNA or on one segment of DNA, there are multiple genes. Okay, and these genes are called as the structural genes. So what we can say that on one segment of DNA or RNA, there are multiple structural genes. Okay, multiple structural genes. And that's why in case of prokaryotes, this segment is called as the polycistronic. When on one segment there are many genes, then that segment is called as the polycistronic. Okay, now we have to see how these genes are getting regulated, and these genes are being regulated by this accessory protein. Clear? So see what points I have told you that in case of prokaryotes, accessory protein is there which regulate the expression of the gene. First point. Then I have told you that in case of prokaryote, there is one segment of DNA or RNA, and that segment of DNA or RNA contains many structural genes, and it is called as polycistronic. That's why it is called as polycistronic, because on one segment there are many genes. Okay, and the expression of all these genes are regulated by this accessory protein. Along with the accessory protein, we have one more thing, that is operator gene, promoter gene. Okay, so see on one segment of the DNA, what we can find in case of prokaryotes. See what I am telling you, that suppose this is one segment of DNA, and on one segment of DNA there are many structural genes. Now suppose all these are the structural genes, and along with the structural gene it also have accessory protein like accessory gene, okay, which regulate. After that it has one promoter gene and it has one operator gene. Okay, so this operator and promoter, they used to regulate the expression of the gene, and everything is controlled by the accessory protein. So what things are there? There is one accessory protein, one promoter gene, one operator gene, and many structural gene. So all these things together, they used to regulate the expression of the gene in case of prokaryotes. And this complete segment is called as operon. Okay, so understood what I mean to say that? I just wanted to tell you that in case of prokaryotes, on one segment of the gene, there are many things. Now what many things are there? There are many structural genes. Along with the structural gene, there are accessory protein. Okay, what is the function of the accessory protein? This accessory protein will control everything. Okay, then there is one promoter gene and one operator gene. And as the name it is clear, the promoter will promote everything and operator will operate the function of the gene. Okay, so promoter is there, so it will promote the structure of the gene, promote the function of the gene, and operator will operate the gene, so that the gene can perform they can form the specific protein. Clear? So what is the function of this two, promoter and operator? That is, they used to regulate the function of the gene, and each and everything is controlled by the accessory protein. Now this complete segment is called as an operon. Okay, so in case of prokaryotes, we have to discuss the operon and the function of the operon. Now, one such important operon is Lac Operon, and this Lac Operon is in our course. Okay, second type is tryptophan operon. This is not in the course, so we will not see this one. Okay, so simply we have to see Lac Operon in detail. Okay, so begin with the Lac Operon. Now see the meaning of operon once again. What I told you, that it is a segment of DNA where many structural genes along with accessory protein, promoter gene and operator gene are present. Then that segment is called as Operon. Now these are the structural gene. Okay. Now this is our operon. Now this operon can be of two types. One is it is inducible and second one is suppressible. Now the meaning of inducible. Inducible. We can induce something. That is, it normally remains off, but it can be turned on. And one such example of inducible operon is Lac Operon that we have just now seen. Okay. And second one is suppressible. I told you that we have another type that is called as the tryptophan. Now tryptophan will come under the suppressible. The meaning of suppressible means we can suppress something. That is, in normal situation, it remains on, but we can turn it off. So this is the basic concept of the operon. Now, we have to see about the Lac Operon. This Lac Operon is normally called as lactose operon. It means in case of bacteria, if the function of the bacteria is regulated by some lactose, it is regulated by a gene which takes the lactose, and with the help of that lactose, they help in the synthesis. Then that is called as the Lac Operon. So, in case of Lac Operon, lactose should be there. So, the name is Lac Operon. Okay. Now, this Lac Operon was explained by Jacob and Monod. So, we will begin with the Lac Operon.